Hi everyone, my name is Licia. I'm really excited to be here today and to talk a bit about my PhD work, which was the development of an experimental platform for automation of optogenetic targeting. So we call it a bit smart microscopy because it's smart microscopes that decide themselves what the next steps in the experiments do and kind of go a bit beyond the traditional automation that you would expect from a normal microscope. Thank you so much, everyone, for inviting me. I hope it will be interesting to you. And there's a bunch of stuff I want to show you from a bit of biology side and also a bit of method development. The platform I already talked about a little bit, but then also have a look more deeply into other smart microscopy applications and challenges, and also have a bit of a look into the future, what there might be. First, a bit of background of what we're interested in the lab. So we're interested in cellular dynamics. We're interested in it at different scales. So we're interested, for example, in cell migration in road GTPases. How do road GTPases self-organize within single cells to enable persistent migration, as for example in this fibroblast, but also at larger scales, as you can see here in this tissue of human breast cancer cells. How do these cells talk with each other? What are the collective dynamics? Here you can see them beautifully moving together like a flock of birds in the sky. So we're interested in how these dynamics relate to cellular fates like survival or cell death. All these kind of dynamic analysis require a lot of data. So here you can see a stem cell colony. We do a lot of image analysis, of course. We do cell segmentation, cell tracking, biosensor readout, anything to kind of automate the analysis of these terabytes of data. And we try to be interactive with it. So we build these kind of small Napari plugins that allow us to have visual feedback of if the analysis actually worked and if we get what we expect from the data. When I came into the lab, the classical workflow is, of course, that you have to experiment. Then you do the analysis after the experiment, you think about the results, and then you plan the next experiment. And the goal of my PhD was to kind of bring this analysis already to the experiment so that we can know in real time what are the data that we're going to extract. And not just after the experiment, but already during the experiment. And this gives us two advantages. One is that we decrease the iteration time because we don't have to do any post-processing of the data. But the interesting aspect is, of course, that we have the data available during the experiment to steer the experiment, so we can use it to control the microscope also in a clever way, in a feedback way that actually responds to the experiment. To give a small example of what we're interested in, when you have a wound in your finger, for example, you get a cut, growth factors are released and distributed to the tissue, and they form a growth factor gradient, and cells are able to smell this growth factor gradient like a dog would smell a scent trail, and they're able to migrate towards the steeper part of this gradient. So they sense the extracellular output, they convert it with signal processing within, and they form a cellular response. Because these cells are so good at processing information, we sometimes compare them to biological mini computers in the lab. So we think about them as signal processing units that have inputs and outputs, and within they have complex signal processing that we try to understand. And we systematically probe the signal processing with optogenetics as input and biosensors as output, as readout. So optogenetics means we can control cellular processes with light. We can activate them extremely precisely in space and time, like you would be able to control cellular behavior with drugs, but instead of adding a drug to your sample and it activates all the cells, you're able to really activate very small parts of subcellular regions of single cells, and also with second control. And biosensors is classical fluorescent biosensors that we use to read out the different responses of the cells. So back to the wound healing example. For example, we can have an optogenetic tool that mimics this growth factor input for cells. So cells feel like they can feel a growth factor input. And when I shine light at the front of a cell, the cell actually starts migrating towards the light spot like a cat following a laser pointer, because the cell thinks it can smell a growth factor gradient, right? A big difference between the electronical circuits and the biological circuits is, of course, that every electronic circuit is exactly the same, but there's extreme heterogeneity in the cellular system. So no two cells look alike. So we need a lot of high throughput to kind of figure out this heterogeneity. And it's not just that there's heterogeneity, but sometimes there's really opposing behaviors. So for example, these two cells have exactly the same optogenetic biosensor circuit. And the cell on the left, I've already shown you, starts migrating towards the growth factor gradient, but the cell on the right does exactly the opposite. And when I shine light at the top of the cell, it starts polarizing downwards and does exactly the opposite of what we would expect. So we don't only need hundreds of cells that we can average, but we actually need the single cell resolution to figure out this kind of heterogeneity. We cannot just average. So what we would want is that we can control hundreds of cells in parallel across multiple field of view so that we can try out different drug perturbations, for example, and have enough resolution to see what the single cell is doing. This gets really tiresome because these experiments can take for days and they're super slowly moving cells, and we need to image them for a long time with time-lapse imaging. And also there's thousands of cells in parallel, so if you would want to do this manually, you would have to sit in front of the microscope and click on the individual cells every couple of minutes across thousands of cells across multiple days, which would be really annoying to do. So the goal of my PhD was to build this kind of robot that is able to shine the light automatically in front of each cell. And the way we do this is that we have this feedback control loop where we have a control algorithm that decides where we want to shine the light. We automatically shine the light on top of the cells, and then we use a real-time image analysis and automated tracking to inform the control algorithm where the cells actually are. In a bit more detail, we use the Micromanager software, which is this framework that has tons of drivers for a lot of the popular microscope hardware. So there's stages, cameras, light sources. There's a good chance that your microscope is also compatible with this. And then we use a Python interface, PyMcore Plus, on top of that, so that we can implement all our control and image segmentation strategies in Python, where it's much easier to do. And as interface, we use Jupyter notebooks. I will talk about this a bit more in detail later, and Napari as graphical user interface that allows us to actually see the different positions and preview the segmentation and things like this. Now, this system really gives us unprecedented control over cellular behavior. Here you can see an experiment where we control hundreds of cells in 24 field of views. I show only the best ones here. Across several days, and we can really control of where the cells migrate to reach this kind of target shape in the end. And each cell is really controlled individually. So you can see it in more detail here with some cells cropped in, and you can see how we can really steer them around, and the algorithm tries to find open spaces on the target shape and steer the cells towards this place so that the shape in the end is optimally filled out. What's nice is that we still have subcellular resolution, of course, and we can do these kind of drug perturbations also. So here you can see an experiment where we just migrate the cell upwards, but with different drugs on top. So white compound, but also pictilisib here, a PI3K inhibitor. And we can do these large-scale screenings of hundreds of cells with single-cell resolution, while also having the cytoskeletal information by looking at the cells and being able to read out quantitative information like migration speed. And this experiment was super cool, and we saw that pictilisib actually didn't decrease the migration speed compared to control, which is really weird because PI3K is supposed to be really important in gradient formation and cell migration. So it's something interesting that we're trying to follow up in current work. As promised, a bit more detail about the Jupyter notebooks that we use to set up the experiments. So it's a very modular pipeline. You can choose different segmentation tools, you can choose different tracking tools, you can choose which channels you want to stimulate. Then we use Napari to find good stage positions. We read them into the notebook. We preview all the processing steps, the segmentation, a preview of the stimulation regime, and then we run the experiment from there. And what's really nice is that this is like a lab notebook. You get saved Jupyter notebook for every experiment you do, and you know all the parameters you've set and all the functions you've used for free. And then what you get as output is a large data frame with all the tracking information, that biosensor quantification, and of course the images and stimulation masks, which you can really easily display in Apari and even overlay the masks and the tracks at the same time, which is really rich feedback that you get already during the experiment. These pipelines are very complex, so there's a lot that can go wrong. At the beginning of my PhD, I spent a lot of time going to the lab in the middle of the night trying to fix some bugs because it crashed like after 10 hours in or something, because there was something weird that we did not expect. So what was really helpful is to have this kind of virtual microscope that allow you to really probe your smart microscopy workflow in silico. So instead of classical simulation devices that read out pre-acquired data and display those, these are actually simulations that respond to input also, so you can really test if the feedback component of the pipeline also works. So here you can see a simulation of cells that behave in a similar way like the cells I've shown you that migrate towards the stimulation regime. And something that was really unexpected is that having this kind of simulation platform that is, I mean, of course, it's not biologically accurate and we don't follow like physical models here. It's really just coded together so that it looks and behaves in a way that we would expect from our sample also. But it's really not physically accurate. But still, it was good enough that we can actually kind of fine-tune these stimulation rules. So for the experiment with the letters I've already shown before, I've used this simulation in silico to kind of go over the different rules and increase the complexity of the rules over time. So for example, in the beginning, I only steered towards the closest place, but then I saw, okay, this crowd cell in a location. So I tried to find like places that are a bit more far apart. And once the shape is filled enough, I steer away the cells so they can kind of clear up the rest of the pattern. So you can kind of iteratively add complexity, and LLMs are extremely good at quickly iterating over this. So you just tell the microscope, okay, steer away the cells once the shape is too crowded. It writes the code, you can test it in silico, and if you're happy, you can go to the microscope and run the actual experiment. So what do we actually use it for? We're interested in, for example, subcellular signal processing. We can test these kind of experiments where we're interested in if there's a difference, if we activate the cell at the top or at the bottom, or if there's some kind of spatial signal processing going on, if the cell senses the differences, if the input is very concentrated in one place, or if the same input is spread across a larger area. So these are kind of things that people have hypothesized with mathematical models for years, but we didn't really have the experimental paradigms to actually test this. When we want to do these experiments where we're interested in the cytoskeletal feedback also on top of the signaling, again, the cells look extremely different. So something that we try to do is that we homogenize the cell shapes using micropatterning. So instead of having cells that all have extremely different shapes, we use micropatterning to homogenize this a bit more so that every cell has a standardized form. For this, we use micropatterning. The classical workflow is that you have microstamps that you can coat with some sticky proteins, for example, fibronectin. You stamp this on top of a glass slide, and then the cell adjusts to these kind of shapes. We didn't invent this, of course. This is a classic technique that is widely used, but we did some work on the protocol. So I tried to kind of repurpose our existing microscopes for optogenetics, but for 3D printing. So you can kind of hack your microscope to work as a 3D printer. Instead of projecting light for the optogenetics, you project UV light that you can then cure. So it's a 3D printing resin from consumer 3D printing that hardens when you shine UV light. Then you can wash away the rest of the resin, and you can do PDM mask masks, which you can use for different applications. We've tested it across scales from 1 micrometer sized pillars and pits up to animal scale for confining C. elegans in the field of view. And then once we have this, we can use computer vision to kick out any cells that, for example, where 2 cells occupy one pattern, and we get a really nice data set with hundreds of cells that have a uniform cell shape, and we can target those with optogenetics. I'll show this later. For the protocol, what's cool is that we were able to replace a bunch of the expensive reagents. So, for example, instead of SU8, which is the classical photoresist, we use off-the-shelf 3D printing resin. We print on microscope slides directly instead of using silicon wafers, and we were able to replace a couple of the toxic chemicals to make the protocol nicer to use. And everything is controlled programmatically from Python. So you're also able to, for example, do exposure test series on a single slide, where you would have to do multiple exposures and to repeat the whole protocol a couple of times with a classical approach. And yeah, I think there's a couple of interesting follow-up questions here. For example, to use the image feedback also to steer the image stimulation intensity to optimize the exposure in real time, like with feedback, for example. Here you can see a microfluidic chip that printed. You can see there's a really micrometer resolution, but also at centimeter scale. So using the microscope stage, we can use a high resolution microscope objective to print one field of view and then move the stage to pattern a whole chip with really centimeter scale chips. Putting together these two methods, the feedback control and the micro-patterning, really allows us to have cells with a specific shape, and then we can really input optogenetic pulses onto specific cytoskeletal structures and measure these input-output relationships while controlling the cell shape also. So in this specific experiment, what was interesting to see is that on the control shape that are around cells where we don't expect a difference, we really get the same response from stimulating at the top and the bottom. But in the cells that have this shape mimicking migrating cells, we see that when we stimulate the top or the bottom where we see small cellular protrusions, we get a much higher ERK response if we input the optogenetic growth factor input. Same for this kind of spatial signal processing. We saw that when we split up the light dose on two different areas in the cell, here in orange, the cell has a much smaller response than if we concentrate the light on the one edge, which means there's some kind of excitability that is very local and spatially controlled. But we also can use it for ROGDBA signaling. So here, for example, is an optogenetic tool where we can activate RAC, so we can activate cellular protrusions. You can see I'm always segmenting the cell and then updating the stimulation mask to make the cell form a protrusion towards the top of the cover slide. And then after some time, I switch a direction and stimulate the right side, and the cell immediately starts responding and starts forming protrusions to the other side. So we can really control cell shape in a specific manner, completely automated with over hours. We can do exactly the opposite here, where we use GEF for row A, so we can activate contractility in this cell using optogenetics. I shine light at the top of the cell, and you can see the cell starts contracting very rapidly in specifically this region. So the first project we've applied this to was a project that was started by a previous PhD student. He was interested in this gap called DLC1 that is known to localize and focal adhesions, and he was interested in if there's a specific subcellular regulation of Rho signaling due to this gap. So what we did is we selected regions on top of focal adhesions and regions between focal adhesions and activated Rho A, and then saw if you can see a different response in those. So again, using computer vision, I virtually split the cell in half, and in the bottom of the cell, I selected focal adhesions, and in the top half of the cell, I selected regions between focal adhesions, and we shine light on those in parallel, and then we see if there's a different response between those. What was really cool to see in this project is that we were interested in how the gaps and the gaps balance the Rho A activity. And what we did first is we did very strong perturbations where we do a lot of contractility in the cell, and we didn't really see a difference between cells that have DLC and cells that don't have DLC, so we did knockout cell lines. But when we started to see differences was when we did these very fine-tuned weak perturbations where we really just nudge the cell out of equilibrium and then saw how it falls back into equilibrium. And this was really only possible because we had this automated way of doing it, and we could screen hundreds of cells and thousands of stimulation regions in a completely automated way. So very quickly, the results that we saw is that in cell lines that don't have DLC1, so the knockout cell lines in pink here, we saw an overshoot of Rowe activity compared to the wild type cells in green. And we also saw a difference on focal adhesions versus non-focal adhesions. So with the same light dose, we were able to recruit Rowe more efficiently on focal adhesions versus bare plasma membrane. Yeah, so this gives you a bit of an overview of this pipeline. We're able to do experiments across different biological scales, so from extremely small subcellular scales, focal adhesions, to cellular scale where we can control cell migration, but also collective scales. So for example, here I have a tissue of scale. So for example, here I have a tissue of breast cells and I shine UV light on a specific cell. I track it over time and kind of give the tissue a mini sunburn. And then you can see how the rest of the population responds to that. So we're not only able to do experiments with optogenetics, but any kind of light-based perturbation. And also in, we have a preprint out. It's on bioRxiv. All the code is open source and public on our GitHub repository. And I'm really happy that there's already other groups that we used this pipeline on for uncovering the role of SARC and HCK in motility of macrophages. And in another experiment, we looked with a more theoretist group at the lab of Daniel Rivelin in Row A regulation in MDCK giant cells. So it's also really nice to get this kind of quantitative data to fit models too. What's a really interesting challenge in these kind of experiments is that we cannot tune or image segmentation algorithms after the experiment. So we need something that works on live data. And as you know, the cells look different from experiment to experiment. You might have new biosensors or you had drugs that completely changed the morphology. So I really wanted a segmentation tool that allows you to work with live microscopy data. So you can start your experiment, you quickly train an algorithm on the live data and then use that to segment your cells or the cytoskeletal structures that you're interested in this experiment. I also needed a tool that was compatible with our Python-based pipeline that works nicely with an Apari image viewer. And at the time, there wasn't really something available. So I started developing compte, which is a elastic-like interactive segmentation tool. It's completely built in Python. And what it allows you to do is draw small scribbles on top of your sample, as you would know from Elastic. And then you can extremely quickly train a segmentation algorithm based on those scribbles. And it's interactive. It works in 3D. It works with multi-channel data. It works with time series, and it works with a lot of different image behaviors, image types. The way that it works is that instead of classical filter banks like Elastic would use, we use pre-trained convolutional neural networks or also vision transformers to extract features from the image, and then based on those features, we train a small classifier, a cat boost or a random forest model, and we use that to train. So we don't touch the weights of the model, we just use those for feature extraction, and then we only train a small lightweight classifier. To give you an example of how this works, so you can repurpose basically pre-trained networks. So for example, CellPose is a really nice neural network to predict cell masks. Here you can see the base CellPose model that predicts cell shapes, instance masks from an input image. And using compte, we can retrain it within a couple of seconds to instead predict only the nuclei, or only predict the membrane, or only the cytosol, or only the dividing cell in this case. So you're able to repurpose or customize pre-existing models to your specific image analysis task. A big disadvantage is, of course, that you lose the instant segmentation, so you only get class labels and not individual cells. So you get foreground, background, and not cell 1, cell 2, cell 3. Using these vision transformers, we can also extract really high-level semantic information. So here you can see a shark example that master student Roman acquired on his last diving holidays, and we were able to, with a couple of scribbles, train the algorithm to predict the different shark body parts in this video. So this would be something that's really impossible with classical filter banks, but the larger vision transformer models are really are able to capture the semantic information and also predict it in this case. Also works in other animals. Here, for example, you can see mice where we segment the tails and head. Also works for behavior annotation. So here I just annotated 2 frames of an open and a closed eye. And when I blink, you can see that the algorithm is able to detect those. So yeah, there's a lot of applications we developed originally for feedback microscopy, but it turned out to work so well that we put it in a standalone tool and decided to release it for the larger community. We spent a lot of time also writing documentation and making it very usable and fast. It runs on laptops very easily. It's called ConfPaint. You can easily install it with pip install ConfPaint. There's a GitHub, and there's a lot of people involved, especially Roman did a lot of work on this and is handling at the moment all the bug fixes and community issues and tries to spread this a bit more. But yeah, I think it's a nice tool, and I think there's a lot of applications where it might work, so definitely try this out. All right, in this last section of the talk, I want to take a step back and look a bit more general at smart microscopy and what the challenges are, especially on a technical level. Smart microscopy, I refer to any kind of feedback loop where we use a real-time image analysis and use this to control the microscope in a clever way. So some of the challenges I ran into is that there's really no standard for hardware microscope control. So whenever you want to transport one workflow to another microscope, you have to basically reprogram it. Even taking it from one Nikon microscope to another microscope within the lab, that just had a bunch of different drivers and different attachments. Took me like 3 months to adjust. So I don't want to imagine how long it will take to take it to a completely different system. Which leads to a lot of incompatible software and also a lot of redundant development efforts across different labs. So I'm really excited that there's the Smart Microscopy Working Group that brings together people from industry, but also different labs to kind of think about how we can standardize these kind of workflows and come up with common APIs that allow to transport these workflows from one microscope to another. We've recently published a paper called Smart Microscopy Current Implementations and the Roadmap for Interoperability, where we tackled exactly this issue and come up with a plan of how we could achieve this. We started by collecting different smart microscopy implementations across industry and academia, and we collected them on the webpage. Every application got its own entry with a standardized setup of the document. So there's a lot of practical tips of how you can actually run this software, what are the challenges, and also with a focus on bottlenecks and interoperability and code repositories, which is really important. Because I guess most of the time when people develop these kind of workflows and publish them in biological journals, they're kind of hidden in the method section, and it's not the main focus of the paper. So the code and the implementation and how to run these things are hidden most of the time. So I think this is a really nice resource to focus on the actual technical implementation. The repository is community editable, so you can just do a pull request in GitHub if you want to add your own implementation, or you can send me an email, and I'm really happy to add your stuff there if you're interested in featuring your software or your hardware. We started by analyzing how different labs implement to bring their smart microscopy workflow onto different microscopes, and there's different layers where you can do this. For example, atomic tools writes this kind of adapters between the smart microscopy software and the microscope control software, which requires you to write an adapter for every specific microscope, and the maintenance effort lies with the academic labs that want to implement it. Another way is the micromanager approach, where you define a hardware interface for all the device types. So for example, if you have a core lab stage, you can control it with the same commands as you would an icon stage. The issue there is a bit that it's limiting the functionality a bit. So if a microscope company wants to implement a specific functionality, they still have to adhere to this common device interface, and they cannot optimize a lot. And a lot of companies also don't want to give up this low-level device control. And the last way that we're kind of trying to promote at the moment is to have an abstract interface where you define a common device API. So you can think of this like image metadata, but before you take the image. So you tell the microscope, I want to take an image with this channel at XYZ position, and then the microscope control software is able to implement these experiment descriptions into actual hardware commands. One proposal for this is the USEC schema, which is a schema that describes these kind of imaging events, and then the microscope control software can implement this, and you can run an experiment in one microscope or the other, and it would just work. More general, we come up with this interoperable ecosystem where we have open data formats, so USEC schema from the feedback control software to the microscope hardware. And the other way around, we pass back OVSAR images, so open data and metadata formats in both directions. Then you will be able to plug and play different microscope hardware, but also additional microscope components. And on the software side, also, it would be really nice that it's modular, so you can plug in different segmentation tools, different tracking tools, or different applications for your smart microscopy workflow. So if you want to detect a rare event, or if you want to optimize exposure in real time, or control cellular behavior. On the user interface side, also, I guess a lot of the tools still use code, which is very flexible, but it also requires people to know how to program. Another approach that I'm really interested in at the moment is also to use natural language as an interface. We'll talk more about that a bit. If you're interested in this, the paper is out. It's also kind of a review of current implementations of smart microscopes and different applications. We focus a lot of the technical implementations and address some of the challenges that cultural and technical, the tension between academia and industry, and also funding agencies, etc. Yeah, I think the Smart Microscopy Working Group is cool because it brings together exactly academia and industry, and we try to kind of bridge this gap between specialized applications in the lab, but also general use cases and the requirements of industry. And if you're interested in this, the group is always excited to welcome new members from academia and industry to join monthly discussions and join discussions of how we can create these kind of standards. Yeah, as I said, one of the last parts that I want to talk about is this natural language interface. With LLMs, we can just talk to the microscope basically and tell it what to do, and then the LLM translates this into specific code snippets that can be executed on the microscope. This project in the lab, we call it the ChatGPT microscope, which would allow you to talk in natural language to your microscope basically, and it runs your experiment. So the way you can imagine this is that you could say, for example, you want to acquire a 1-minute 20-frame time-lapse in the UV channel, and then the LLM translates this into the specific API call. Here you can see from pre-training it inferred automatically that by UV channel, I probably mean the DAPI channel. So by pre-training and also having access to the microscope configuration, kind of is able to adjust to support you with controlling this complicated hardware. So you can imagine it like having an experienced microscopist next to you that has knowledge of the exact microscope, your cell lines, etc., and is able to translate your experiment idea into concrete commands. But then the next step would be to use LLM also as a controller, to not only use it for automation, but to also use it as a more autonomous decision-making software. In robotics currently, this is state-of-the-art in high-level planning and then breaking this down into smaller subparts to achieve a really complex behavior from natural language description. I show you some very preliminary results. These are kind of old. I should update these slides. But here you can see that I asked the microscope what channels are available. It's able to read the microscope configuration and gives me a list of the available channels. So it really has access to your specific configuration. And then I ask it to change the camera binning from 1x to 4x and calculate the mean intensity. And the LLM is able to come up with code that does exactly this and then follows up also with the image analysis and the data analysis and presents the. So here it's running on a virtual microscope. The API is exactly the same on a real microscope. We test this. It's also working. I'm moving a bunch of windows around here. It's a bit messy, I'm sorry, but you can see the binning and there in the window change from 1x to 4x. You can see that the image that is being displayed gets much brighter in a second, and then calculates the mean intensity of both these images and gives me back the results. Here's another example where we ask the microscope to take an image and then segment the cells. So this is again running on the virtual microscope simulation. We just tell it to take an image and segment the cells. It's able to first write the code to acquire the image, but then also write the code to segment the cells. And then by giving it access to the Napari interface, it can also directly overlay the masks that it calculated on top of the microscope image to give you very fast feedback on that. You can see the masks appearing in a second, I guess. Let's see. There they are. It works. I think there's a very clear path on how to implement this. So first level, it would be like chatting. So it would be like a chatbot that knows the microscope documentation that has access to the specific hardware that you currently run. I think we've managed this. And currently we're working on solidifying the linear code execution. So straightforward experiments. Bringing towards the level 3 would be feedback so that you actually iterate on the experiment. And I think other fields, for example, chemistry is already at level 3 and robotics is way ahead of us, going from really high level task descriptions to completely autonomous implementation of that plan. And I think microscopes are really just imaging robots, and there's not really something that's limiting us from also implementing this on microscope. To give a bit of an analogy, maybe you know those AA apps where you can take a picture of your fridge and tell the LLM, okay, you want to eat something healthy with the ingredients you have available in your fridge, and then the LLM comes up with some kind of recipe like, okay, if you have cucumbers and yogurt, you made a tzatziki. So why not to take a picture of your -80 and say, I want to do these and these experiments, and the LLM will come up with a plan which cell line to use and automatically runs the experiment. This sounds a bit crazy now, but I think they won't take too long until this will become a major part of how research is conducted. Yeah, I want to end up by saying thank you first to all the master students that contributed a ton to this project. Joel, Remo, and Moritz did a lot of the microfabrication protocol establishing and also did a lot of the experiments for the map-based stuff. Roman is working on ConfPaint and Dario is doing the LLM control. Roman and Dario now both have finished their masters and are doing a research internship in the lab. Also, I want to thank the whole lab, especially, of course, Olivier Pertz, which is a fantastic PI. It was a ton of fun to do this PhD. I got a lot of freedom to explore and to follow these kind of crazy ideas that turned out to work some of the time. Yeah, but it was super exciting and a lot of fun. And I'm going to leave you with an overview of the manuscripts that I presented in this talk in case you want to follow up with reading a bit more. Most of these also have GitHub repositories with documentation, etc. I'm pretty active on Blue Sky, so I post a lot there. I'm happy to connect there, but also feel free to send me emails, of course. And yeah, I plan to move to Paris after this postdoc for another postdoc. So I'm really excited specifically to connect also with the French bioimaging and smart microscopy community in particular. Yeah, I hope that was fun and was interesting to you. And yeah, happy to get questions or connect in any other way. Thank you so much. Bye.